Hi, thanks very much. I'm going to give a very basic talk because I've discovered on this topic that while there's a huge safety database on most of these sweeteners, it's rather old. And so when the current concerns in the public about the safety of low-calorie sweeteners, almost nobody is aware of this large trove of information. So it seems appropriate to kind of reintroduce it to people so that you can use it as a backdrop of information when you're confronted in the public by these concerns about the safety of low-calorie sweeteners. So that's my intent here. And it's nice that we have a discussion this afternoon because this is very basic information, since I don't know where you begin in your knowledge of this topic. But there are loads of questions. And I'm just touching the tip of the iceberg with this information. There are a lot of interesting things to talk about. Okay, so by way of introduction, low-calorie sweeteners have been around for over 100 years. The first one was saccharin. My interest in this started almost 40 years ago as a side interest of research I was doing on amino acids and brain function, because in the United States, both cyclamate and saccharin had been banned. Those were the only two low-calorie sweeteners at the time. And then aspartame was coming on the scene, and this occurred right at a time when the Congress had decided they wanted all food additives reviewed for safety. So compounds, especially the compounds that were designated GRAS, which means generally recognized as safe, they wanted scientific panel to go through and review the safety literature on all these compounds. So aspartame kind of came in for review at a time when all of this was going on. And there was an extraordinary amount of attention paid to it, both by the regulatory group, the Food and Drug Administration, and by people in the public who decided to make an issue out of it. And as a result of sort of a several-year controversy, the company that developed aspartame spent a very large amount of money doing a large number of additional animal and clinical studies to demonstrate safety. In addition to the submission to the Food and Drug Administration, which was quite extensive. Food and Drug Administration food additive submissions really are quite extensive documents in terms of safety information. Food and Drug Administration in our country, in regard to food additives, they're not looking at functionality, they're looking at safety, because the products that they approve go into society in a totally unregulated fashion. So anybody can use an approved food additive or non-food additive any way they like. So when they approve a food additive, they want to make sure that pretty much the intended use and their calculated level of exposure will basically provide a safe level of intake for anybody who chooses to use it, and pretty much at any level of intake. So that's kind of the backdrop to this. Let's see. There we go. So what I'm going to basically discuss is just give you an idea what the low-calorie sweeteners are, how do they work to generate sweetness. There's a new literature on this. It's quite exciting for sweet taste in general. Are they safe? I'll give you a basic idea of what the expectations are by our Food and Drug Administration, also EFSA, the European Food Safety Authority, what their expectations are in terms of what you have to submit to them to give them some belief that what you have is actually a safe compound. It's very useful to know how much we eat, because in all of the stuff that's going on in the public, and it's not just here, it's also in the States and in Europe and elsewhere, you need to have an idea of how much you're actually eating in relation to what the authorities tell you is a perfectly safe level to eat, to be able to reflect on some of the opinions that are flying around. And the current concern, the one that's most in the public these days, is do they make us eat more and make us fat, which is especially surprising to me, because that was a key issue 40 years ago, which was totally resolved, but now it's back and everybody hasn't learned or has forgotten that all this information exists. The short answer is no. So just to begin, here is a slide that just shows you there are many different low-calorie sweeteners. They are very different in terms of their structures. Sucralose, for example, is a sucrose with molecule with a couple of chlorides hanging off of it. Saccharin, ACE-K are small heterocyclic molecules. Aspartame, that's the one I just mentioned, is a dipeptide discovered accidentally while Searle, a drug company, was looking for new ways to suppress acid secretion so that people with ulcers wouldn't feel so bad. This is the terminal two amino acids of gastrin and cholecystokinin, two gut hormones that influence acid secretion, promote acid secretion in the stomach. So it wouldn't be surprising they'd be looking for peptide pieces in these molecules as inhibitors of acid secretion. Quite accidentally, they tasted one of the things they were testing, and it was intensely sweet. Totally random. That's how aspartame entered the universe of sweetness. And then we have recently, and this is just one molecule, but it's in the food supply now, stevia riboside A, a stevioglycoside with glucose moieties hanging off the side, quite different in structure from all of these molecules. And then, believe it or not, small proteins from plants, like brazane here, 54 amino acids, this one, that can be intensely sweet. There are some proteins that have been discovered in plants that are up to 100,000 times sweeter than sugar when comparisons are made. Recently, aspartame's been quite an interesting molecule because different things have been hung onto it to see if sweetness would change, and the latest version, which was just approved by the US FDA, is a version of aspartame that has a vanillin residue. That's what makes vanilla taste like vanilla, off of the aspartate portion of this molecule, and it's some 20,000 times sweeter than sugar. So it's 100 times sweeter than aspartame. The FDA now doesn't worry about phenylalanine intake from the sweetener because you lose so little of it. The amount of phenylalanine you end up ingesting is trivial, not important. So I'll come back to this in the talk later on. So anyway, so you can see there are a variety of molecules, very different in structure. Folks who worked in this area in the 80s and 90s thought there's such a diversity in sweet-tasting molecules, there must be multiple receptors on the tongue for it. It turns out that that's not true. Thanks to molecular biology coming to taste physiology in the late 90s, the sweet taste receptor was identified, and it seems to be there's only one of them. And it's a structure that is two proteins. The VFT here, the structure of the taste receptor, two proteins very similar in structure. In the portion that sticks outside of the cell membrane is called a venous flytrap. This is where a common structure for receptors where the sweet molecule in this case binds to that portion of the receptor and begins the cascade that leads ultimately to your perception of the sweet taste. It's anchored to the cell membrane by something called the transmembrane domain. In this case, it has 7 protein components, and then on the inside, it ends at the C-terminus, and this is the point where the intracellular signaling molecules are generated. In this case, this is called a G-protein coupled receptor, and so there are G proteins that are sitting here that are activated to release intracellular signals when this venous flytrap domain is hit by a sweet molecule. The portion on the other side, this other protein, I'll just say for the sake of this discussion, I'll leave alone. There are a lot of interesting features about it, but generally I don't want to spend time on it. I will say, however, that using molecular and cellular techniques over the last 10 years, the interaction of all the sweeteners that I've described, plus many others, with this sweet taste receptor has shown that they all interact with it and they all generate sweet taste through this one receptor. And interestingly enough, they all bind to different parts of the sweet taste receptor. So there's, there's the protein cyclamate, one that we aren't allowed to use in the U.S. but is in Europe. Lactazole is a molecule that blocks sweet taste. They bind to this other protein, which I won't go into. Aspartame, glucose, sucrose, and sucralose, all monosaccharide, disaccharide structures bind to the Venus flytrap. And the others that have been studied also bind here in different places. So that's really quite a surprise. And it's really a tour de force in molecular and cellular biology that demonstrated this. I won't go into this, but it's just, I think, a fun thing to know. This is an area in the process of very rapid development, taste physiology, especially with the discovery of this and related receptors. Okay, so in beginning to talk about the safety issue, the first thing I think is useful for you to know is how much of this stuff do you eat, and how much do you eat in a population compared to how much that a regulatory body says is okay to eat. So I'm going to describe to you kind of what our Food and Drug Administration and also EFSA does in terms of requirements to demonstrate safety. But one number that they are particularly keen on is something called the ADI, acceptable daily intake. How much, here's the definition here, it's a level intake by humans that may safely be consumed every day for a lifetime by virtually any member of the society, and this means, you know, practically newborns all the way to old people with pretty much any disease you can imagine, without any health or safety concerns. Okay, it's generated from data that are submitted in what's called a food additive petition, animal data that allow them to set this number. It's based on pharmacologic and toxicologic principles, but this is a number the FDA generates, in terms of daily intakes, these two columns here, when you submit this petition, you also have to estimate for your compound, what do you think the population will eat, right? So that they have some idea that they can reflect off of this ADI they've determined. So what you typically do, let me just say specifically for aspartame, which is the most studied low-calorie sweetener, period, and probably the most studied food additive in the U.S. history anyway, that the estimation was made like this. Okay, so if we look at the average American diet using NHANES data from national surveys, let's make our first guess. All the sugar, added sugar that's put in the diet will be replaced by aspartame. And if you do that, you get a number that's about 8 milligrams per kilogram per day. That's pretty high, all the sugar that's added. You would, in practice, you wouldn't see that, right? So that's a high estimate. If you take and calculate for extreme users, well, actually, if you take and calculate if all of the carbohydrates in the diet were replaced by aspartame, so this would be roughly 40 to 50% of the total caloric intake per day, you get to a number that's about 25 milligrams per kilogram. Okay, now that would never happen in practice, but you're trying to go, here's an upper limit. It's a ridiculously high upper limit based on what people normally eat in the way of sugar and total carbohydrates. So you can see that a number of 8 and 25 is well below this predicted value for safety by the FDA. So the FDA looks at that and goes, fine, no problem. Then the additive gets approved. Now you come back and they require you to actually go out and monitor intake in society, in the population, after it's been approved. So what happened in the case of aspartame was the FDA said, you specifically have to go out and hire someone to go get a giant database and look at what the actual use is over a period of years. So they did. And that's where these numbers come. These are quite accurate numbers for aspartame, less than 5 milligrams per kilogram per day. And for the highest level of intake shown from that database, about 13 milligrams per kilogram per day. So you can see that the actual average use is less than a tenth of what the safe ADI is that the FDA had determined. You can see for the other sweeteners, I just picked out 4 that are common, most common in the U.S. Marketplace: saccharin, sucralose, ACE-K, and aspartame. And you can see they're all, average intakes are all well below what the FDA has designated as the ADI. Keep in mind the ADI is determined not from the highest level, a level that has gotten to in animal studies where a toxic effect has occurred. It generally turns out to be the case for these compounds. It's the highest level that an animal would tolerate. The reason most likely it's higher in the case of aspartame is rats can't taste aspartame as sweet. And as a result, you can keep piling it into the diet and they're perfectly happy to eat it. But in a case like sucralose, for example, which rats can taste as sweet, you get to a high enough level and they just go, had enough. And so you see a reduction in food intake. And so then you'd come back to the company and go, oh, is there a toxicity? Rats are eating less at this high level of intake. So they had to go show that no, it had nothing to do with that. They ate less because it was simply too sweet. So these numbers are actually numbers generated by a no effect level. So the actual level that might cause some toxicity would be quite a bit higher if it occurred at all. So here's basically the list. You can see for submitting a food additive petition, gene toxicity, metabolism, pharmacokinetic studies, toxicity short-term in rats, about 90-day toxicity studies, non-rodent species, which would be dogs and primates in some cases, as it was in the case of aspartame, reproduction studies, 1-year tox tests with non-rodents. This is basically putting different levels in the diet and feeding it to animals for a year and looking to see what are the common things that you would investigate if a toxicity was suspected of being present. Chronic lifetime studies that look at tox and carcinogenicity markers, and for anything that got approved, all of these turned out to be negative. In addition, and important, is as the FDA goes along reviewing this pile of data, it gets questions, asks for issues to be proposed, and then it'll go back to the company and say, oh, why don't you go do some additional studies like this, this, or this. And I pick out aspartame purely because in its case, the company was rather stubborn about it. They would go and do every single study based on what the FDA was asking them about. So there's a rich literature, scientific literature, primary literature on this. This is my favorite summary of this literature for anybody that's interested to look at it. But you can go through and just see all of the data reviewed. Most of this has to do with the additional studies they did, mostly in humans, that were asked for by the FDA. I'm not going to go into this. This is actually a fairly colorful history that's fun to talk about, but I just wanted to hit you with this from the standpoint that there are piles and piles of basic data generating studies in animals and humans in addition to everything the FDA requires for this particular sweetener. So for people to ask these days, is this stuff safe? Are there any data showing safety? It's kind of a ridiculous question because there's just enormous amounts of information. Sucralose and ACE-K went through similar reviews much later, but also in the published literature there is quite a lot of data that you could look at if you wanted to be convinced at that level. So the latest thing is, do low-calorie sweeteners make you fat? And I personally put that in a safety issue. And this is something that's kind of popped up, largely because of epidemiologic studies, which means I went out and got 5,000 people. I asked them if they ate any low-calorie sweeteners. For those of you who don't know, humans don't usually report accurately on what they eat. There's plenty of studies to show that. But you have to have some basis. So assessing what people consume from records that they submit is pretty much what the standard is. So epidemiologists take data like that and then do things like weigh you and look at how your weight changes over time, and they generate correlations. And one correlation that's been generated is, for example, a study like this, which is from the San Antonio Heart Study. San Antonio's in Texas. Obesity is a big problem, especially in San Antonio. And so they collected information on reported sweetener use at a time that they began to follow body weight. And since this was 1979 to 88, this would have been largely saccharin, not any of the newer sweeteners. And so they found a correlation. Okay, if folks reported eating more, they tended to weigh more. Now, you can look at that and go, all right, the study can't determine causality, which simply means if you find this correlation, it means people ate the sweetener and they got fat, or it means they were fat and thought, wow, I've got to worry about this, so they started eating the low-calorie sweetener to reduce their caloric intake, right? Any epidemiologist knows this and should be careful about how they interpret their data. But conclusions like this, which I think are a bit unfair, really grab the attention of the media. So these findings raise the question whether artificial sweetener use might be fueling rather than fighting our escalating obesity epidemic. And this is 2008, and you'd go, okay, fine, are these people aware there's a large database on this specific issue? If you eat low-calorie sweeteners, do you eat more and gain weight? Right? They weren't. They didn't cite any of this literature that existed. And the one that I think is the easiest one to show you is this amazingly simple study done in 1989, almost 20 years before this Epi study was published, where adult males were taken and given aspartame. You have to give it in pills at this dose because you can't put it in food. Folks don't like it. 1.5 times the ADA. So that's 75 milligrams per kilogram per day, which they took in pill format. That's 15 times the average daily intake, right? 1.5 times the ADI. So it's a huge amount of aspartame for 6 months. And you can pretty much see the result. Placebo or aspartame, the black dots, nobody gained any weight. So if this stuff was magically making you want to eat more and gain weight, you don't see it here. Okay? So that's about as extreme a study as I can find. On the other hand, better studies, or how should we say more appropriate studies, are doing human studies where you're placing the aspartame or whatever sweetener it is. And some of these studies, 2002 and 12, there are several sweeteners that are in use. So you place it in the food supply that these people normally eat, the ones that you're going to examine, and you set up a study, a double-blinded study and so forth, intervention study, in a way where the person is consuming an artificial sweetener, a low-calorie sweetener, in the context of their normal diet. And then you look at them over time and see, does the presence of that low-calorie sweetener in one group versus the other, which doesn't have it, lead to any changes in food intake and body weight? Right? So, you know, I just ordered this. These are the 4 studies I'm most fond of because I'm happy to defend them and feel that I can't really be attacked because they're such well-designed studies. But in the case of Parikas et al., Ted Van Italy was one of the original investigators. They go back to 1976. This is really a very nice study. Then it's only 12 days. Okay, here's one that's 3 weeks, 10 weeks. I've ordered them by length, and then finally 18 months. But I just want to touch on this one for a few minutes. It's the one published in 2002 because it shows some interesting features very clearly. So this study by Ruben et al., it's Arnie Astrup's lab, a Danish group, very, very competent group. They were comparing sucrose with artificial sweeteners to look at ad-lib food intake and body weight over a 10-week period. So they used overweight subjects. They brought them into the clinic, screened them, and then accepted subjects, and then basically sat down with them and go, okay, what do you like to eat in your diet? For each one of them, they determined the things that they like to eat into which they could substitute low-calorie sweetener for sugar. So when they constructed the diet to give these people to eat, they were using familiar foods to these folks. And they'd just taken a sugar-containing yogurt, for example, replaced it with a low-calorie sweetener containing yogurt. All right? They told these people that they were testing different low-calorie sweeteners, not sugar versus a low-calorie sweetener, which is the actual intent of the study. So everybody in the study thought, oh, I'm just getting some foods to eat that have low-calorie sweeteners in them. Okay, so it removes a certain bias should anybody get an idea as to what might be going on. And so what they did then was they said to folks, okay, every week we're going to give you this amount of food and beverages to consume, take them home, eat what we're telling you to eat. It's about 20% of your normal caloric intake on a daily basis, and then just add your normal diet to it, whatever you want, right? So they're not taking the normal diet and adding something on top. They're just saying, here, eat these, and then eat freely whatever you might eat. Okay. And so the standard liquids, beverages, fruit juices, solids, yogurt, marmalade, ice cream, stewed fruits, and other things, okay? Okay, so here's the food log data. with all the caveats offered in terms of how reliable such data are. Bear in mind that what you're seeing is the sucrose group compared to the sweetener group. Both groups comparing data comparable in their characteristics, otherwise than the intervention. So you assume if there's error in reporting, it's on both groups, okay? But look at this. The folks who, this is plotting total daily energy intake in joules, kilojoules. Right, I think in calories, not joules, sorry. But what you can see here is a chart of what they report at week 0, just when they're beginning, at week 5 and week 10 at the end of the study. Carbohydrate intake minus the sucrose in the diet, sucrose, fat, protein, and alcohol. And the first thing to see is in the group that was had sucrose instead of a low-calorie sweetener, over the period of the 10-week period, they increased their intake, total daily intake, pretty much all attributable to that increase in sucrose intake. They didn't sense it was there, pretty much. In the sweetener group, the low-calorie sweetener group, and this would be a blend because they're using natural products from the marketplace. It's ACE-K, cyclamate, and sucralose are the 3 sweeteners. You can see that there's a slight drift downward in their total caloric intake. So number 1, they're not eating more. Okay, so the low-calorie sweeteners put in their diet on purpose is not causing them to eat more food, more calories. And you can see there's a slight reduction in the sweet intake from sucrose. Okay, so number 1, this points out that the low-calorie sweetener is not causing them to eat more food. Now, you don't have to believe that. You can actually go and look at the biochemical, at the physiologic data. So they're measuring body weight, and they're also doing DEXA to get fat mass. And not surprisingly, as they eat more calories on the sucrose over the 10-week period, they gain weight and their fat mass also goes up. And you notice, consistent with the fact that they're eating the same or slightly less calories per day with the low-calorie sweeteners, their body weight is slightly drifting down and their fat mass is slightly but not significantly drifting down. So the food intake data, as reported, is consistent with what the fat mass data show and the body weight data show. Okay, that's a good thing. So finally, let me just point out one other thing here. Is we're told that one of the things we're told now is that low-calorie sweeteners, they're intensely sweet, right? So they tend to make you like sweet taste more and want more sweet-tasting foods. And so that's one reason that they might promote eating more sweet-tasting foods. Sweet usually carries fat, so more calories and you gain weight. But you can see here there's absolutely no indication that the ingestion of the low-calorie sweetener is causing them to seek out more sweet-tasting foods. And I think that's a very interesting feature in this study that no one has really paid attention to. And there are other data as well that show if you're using a low-calorie sweetener, it doesn't make you want to eat more sweet foods. Okay, and that's just one of the studies. The other ones on that list are all interesting in their own way and point out different features of this issue. So just to summarize, I've showed you what some of these low-calorie sweeteners look like. They all act by one receptor, amazingly enough. The low-calorie sweeteners are approved, and this is now U.S. FDA, only after extensive testing for safety. And from my reading of all those data, and I've read a lot of them, they're safe. Okay. EFSA is the same way. The Euro folks are the same as the FDA. They're even better because they actually go and re-review every couple of years. Is there anything new in terms of safety for the sweeteners? Aspartame was last reviewed by them in 2013. As I pointed out, we actually consume very little of the low-calorie sweeteners in our diet. And let me add, only about 30% of the U.S. population touches them. Most people in the population don't care, don't really seek them out. So it's actually a fairly small group of people in the population that use them, and even a smaller one that use them at significant levels. However, the public still believes that they aren't safe, and I think part of that is that they just don't know all of these data exist on safety. And then the latest concern that low-calorie sweeteners make us fat, the data show this is certainly not true. And so we have a conundrum. How do we kind of communicate this information? And it seems folks like us are only beginning to be aware that this is an issue we ought to confront. Thank you.